It's a real pleasure to be presenting my work today. I'd like to thank the sponsors for the conference, especially Illumina for sponsoring this session. And I'm looking forward to talking to you about my project. It's basically about pathway analysis. Now, I'll just give you a bit of a background as to what I mean by this. And if you've been involved in omics analysis previously, we tend to have this flow of large raw data getting summarized down to more and more distilled into meaningful information. So we have sequences that get summarized down to gene counts and further on to differential expression or differential enrichment. Then we want to know which pathways are involved, because we know that the pathways that are involved can give us clues about a mechanism, and that's going to be the main thrust of the papers that we write. Clearly, pathway analysis is a really important and key method in computational biology, with over 13,000 mentions in the literature according to PubMed, and 2,000 just last year. So it's important that we do this process correctly. There are different ways to consider pathways. So you're probably familiar with the literature curated pathways like KEGG and gene ontology and reactome. But we can also consider other types of molecular signatures from MSigDB or harmonisome as kind of data-driven pathways or gene sets. And if you're feeling really adventurous, you can go into these different data warehouses, including Roadmap and Blueprint and ENCODE and many others, to try and curate your own pathways or gene sets to assess a certain question and query your own data in interesting ways. Okay, so when we talk about the methods that pathway analysis is done with, there are three main branches of this. You might be familiar with overrepresentation analysis, which is still the most commonly used method. And it uses something like a Fisher exact test to see whether genes in a particular set or a particular pathway are overrepresented in a differential expression experiment. So there's an arbitrary threshold set, and then we measure that level of enrichment. And yeah, there are a couple of other statistical approaches to do that. The next type of analysis is functional class sorting, which encompasses methods like GSEA and camera and a few others. And it's different because it doesn't involve setting an arbitrary cutoff. So you let rank all of the genes from the most upregulated to the most downregulated, and then ask the question on this scale here, are they bunched up towards one side or the other? And the third branch is pathway topology, which uses most of the concepts from functional class sorting, but uses extra information about the shape of these networks, in particular, the importance of the nodes and the direction of these interactions. So this is theoretically where future development should be put into, but we're really lacking the comprehensiveness of the databases to do this in an effective way. So most of the methods that are being developed are still done around functional class sorting, and that's what I'm going to talk about today. So how was this done? We begin with some omics data. On the left here, I've got some RNA-seq expression data differential. So we're looking at one group of samples against another. We've got gene accessions, fold changes, and p-values, and we've got some gene sets over here. So first step is to assign each of these genes a differential expression metric. And in this case, the upstream tool, DE-seq, has provided us a test statistic, so we can do that. And then we can perform ranking of this gene expression profile on this. The next part is to make sure that these gene accession numbers can talk to these gene symbols, so we need to associate them with a library. And then we can intersect with these gene symbols with the sets and see whether there's some sort of enrichment score. So we have FCS algorithm like GSEA, there are a few different other ones, and then we end up with some sort of enrichment profile. And if this was a null enrichment here, then these points would be kind of sprinkled across the whole ranking here. But as you can see, they're definitely bunched up towards the downregulated side. Okay, so I'm going to talk now about how the Mitch bioconductor package addresses this problem and then takes it to the next level. The package uses the univariate approach of calculating the enrichment by the one-way ANOVA on ranks. And another way to say it is the Kruskal-Wallis H-Test. And that's why this enrichment analysis yields ANOVA p-values. The reason we use this is because it can be readily extended to the multivariate case. So what this means is you can provide multiple rankings into this tool, and it will start to solve this in 2 dimensions, 3 dimensions, or even more. So let's take an example of a 2-dimensional gene expression study here. So what we've got is data from a study of hepatocytes that have been stimulated with high glucose, and the high glucose stimulation is represented on the x-axis, and also response to valproic acid. So that's used in the clinic, and it's a HDAC inhibitor, and we're interested as to whether it can kind of switch off any genes that are responsive to hyperglycemia. So genes that will be upregulated by VPA will be up here and downregulated in this region there. And what Mitch really seeks to do is to identify gene sets that deviate strongly from the rank density plot of all genes. So here we have all of the genes represented in this panel here, and the darkness of the patch indicates how many genes are in that neighborhood, in that 2-dimensional space. Now, Mitch here has identified one gene set, regulation of the complement cascade, which has a enrichment towards the bottom right of the chart. So what this means is it's upregulated by hyperglycemia and downregulated by valproic acid, which means that it's really interesting from a diabetes research perspective. One of the things that Mitch has to do also is to calculate an effect size. There's a couple of ways to do this, but the simplest one is just to take the Euclidean distance from the center or the origin of this chart down to where the mean rank of the gene set is. So that's around here. So the longer this hypotenuse, the larger we can consider that effect size. Okay, and yeah, this study was published just earlier this year. Okay, so why should we pick Mitch over any other competing gene set analysis tool? The first one is that it's one of the only tools that's natively multi-contrast. It expects that you're looking at multiple contrasts. For example, you might have an experiment with lots of disease subgroups, lots of treatment groups. You might be looking at gene expression, histone mods, DNA methylation, all in the same samples. And so Mitch can accommodate that. It plays nicely with a lot of upstream tools in the Bioconductor ecosystem. So there's not a lot of data wrangling to get your data working in Mitch. It's versatile, so you can use any gene set library. You're not locked into gene ontology or a certain or any other. You can build your own if you like. And in terms of its algorithmic perspective, it's very fast and accurate and has really great visualization options, which I'll show you. And if you're interested in that, the paper was published last year. So I'm going to dig into a specific example for where we've used Mitch to look at dynamic changes in chromatin modification, in particular a exercise study. We were lucky enough to get muscle biopsies from participants before and after a 60 minutes bout of exercise at 70% of their VO2 max. And on these biopsies, we did bulk RNA sequencing as well as ChIP-seq for the acetylation to histone 3 lysine 9 and also histone 3 lysine 36. And we did this because we knew from previous studies done in the labs of Mark Hargraves and Sean McGee that acetylation on a kind of global level is drastically increased by exercise. So we did this in 6 participants. In terms of bioinformatics, we mapped the reads with STAR, the RNA-seq reads, and for ChIP-seq, we mapped with BWA and then counted reads specifically around transcriptional start sites so that we could correlate gene acetylation with RNA expression. We used the paired information to do our differential expression. And if you're interested in running analysis such as this, all of the code to do this is in the GitHub repository, so you can follow along at home if you've got access to RStudio. Okay, so let's first have a look at the differential expression and the differential enrichment in these studies. At the level of RNA expression, we had 667 differentially regulated genes, and there was definitely a bias towards upregulated ones. And if we look at K9 acetylation, we had many more differentially enriched genes. There was over 8,000. So you can also see a very strong bias towards upregulated genes here, which is consistent with the previous work in the field on looking at global acetylation levels. And also for lysine 36 acetylation, there was 3,000 differentially enriched genes with a bias towards downregulation. So what this kind of suggests is that there's really large-scale changes to the acetylation of the histones, but these are yet to appear in manifestly in RNA levels after just 1 hour of exercise. Okay, so in terms of running Mitch, this is kind of how it works. You have a gene set library. This time we're using Reactome, and we start with over 2,000 gene sets. And we had to exclude 1,000 of these because there weren't enough members in the set that were detected. So that leaves us with 1,200 gene sets. And of those, 579 of those gene sets had some enrichment with FDR less than 0.05. So one of the ways I like to present this data is by looking at the enrichment scores in a scatter plot like this, and then we can look at the correlation. Now, for lysine 9 acetylation, we would expect a really nice correlation here, meaning that we'd expect genes to have increasing acetylation at the same time as RNA expression. And that is mostly the case. There were relatively few gene sets with reduced acetylation and expression, but interestingly, we had some gene sets that were discordant. So their acetylation went up, but their RNA expression went down, or vice versa. And we can do this analysis for all of the 3 different types of omics that we've done. Now, I'll just skip forward to the next part, which is how do we go about prioritizing all of these findings? We've got over 500 gene sets that are statistically significant. So how do we interpret all of that huge amount of data? We could start with looking at the gene sets with the smallest p-value. This kind of makes sense, but what we end up with is a set of large sets with kind of small effect sizes, which doesn't make them very interesting. On the other hand, I'd like to suggest that we can first look at the gene sets with the largest effect size. Even though they're relatively small gene sets, maybe they represent a more specific biology. So let's have a look at this exercise data set. And this is a scatter plot of the effect size on the x-axis and the significance on the y. So the most statistically significant finding here was signal transduction. And as you can see, the effect size is relatively small, and the size of the gene set is over 1,400. So maybe it doesn't represent a very specific biology. On the other hand, we have FOXO-mediated transcription of cell cycle genes with only 15 members, which has a really huge effect size, and even that adjusted p-value is respectable as well. So this is probably the way to go for prioritizing your gene set results. One of the nicest visualizations that Mitch provides is a heat map of all of the gene sets with the top effect sizes. So here we have that heat map, and the red indicates upregulation and blue downregulation at RNA expression, lysine 36 acetylation, and lysine 9 acetylation. And what we can see at the top of the heat map is a really strong activation of a really diverse bunch of signaling pathways, from hippo signaling through to AKT, through NGF, MECP2, and things like inflammasomes as well. But we also have interesting discordant regulation of other gene sets that have lower levels of acetylation and increasing expression, or vice versa. So we're still trying to figure out what might be the reasoning behind these interesting observations. Okay, if we're interested in a particular gene set, Mitch allows us to kind of drill in and gives us a report and see what's happening there. So what we've got here is a density plot that's projected in the 2D space. In this case here, RNA and K9 acetylation, and we can see that this gene set, ROR-A, activates gene expression, is highly upregulated in both. Whereas for the lysine 36 acetylation, that patch of enrichment is spread out. So it doesn't look like there's anything interesting happening with lysine 36 acetylation here, in contrast to the other two models that we're looking at. And naturally, everyone wants to dig in a little bit deeper and see which of the genes are driving this enrichment. So Mitch provides the list of genes and their relative ranks in that omics data. So you can design primers and follow it up with specific experiments. Okay, and with that, I think I'll finish. I'd like to thank Anthony Caspi, who co-developed the package with me. And I'd also like to thank the Bioconductor team, especially Kayla Morell, for the code review. I'd like to thank Ishant Karana and Scott Maxwell for early testing of the Mitch package, Mark Richardson for manuscript comments, our compute partners at Nectar. And I'd like to thank Sam Alosta, Hari Krishnan Kapanakal, and Mark Hargraves for access to the exercise dataset. And also thanks for your attention.